So this one we have seen before, right? The protocols and drug discovery, the complex interplay of medical chemistry and biochemistry, and where the drug candidate need to be to discover a drug candidate takes a lot of steps, and which requires a huge amount of investment, including the time. That is $800 million approximately in case of a drug to be discovered, and also it takes 8 to 14 years of time. And the steps including the target identification and understanding the the design and the screening by assay to get some hits, and then we find some in vivo studies to become leads, and the lead optimization and the nomination of the candidate to be a drug will take at the end. So that is not going to be the end. So again, that will enter into the the clinical trials that requires again clinical trial 1, 2, 3, and marketing, and then go for the clinical trial 4. It's a huge amount of time and investment. So wherever you can able to reduce the time using any technique, it's going to be very beneficial to the pharmaceutical industry and the society. So this molecular docking is helping in this drug discovery at the various steps as we have discussed earlier. And one of the steps is to understand the protein or the target and your possible drug interaction. So that if you can able to roughly identify the interaction instead of a full identification. So that estimation can reduce the time period, which in turns can reduce the cost or the investment on it. Target identification is the which protein is vital. Okay. So we need to first understand that the what is the protein we are targeting, and then lead identification, pharmacology and the patents point of view, and lead optimization, optimizing, improving the potency. So all these areas, our molecular modeling can help. So in the medicinal chemistry, where traditional screening were actually like we don't know actually what is the target and what is the drug. Just go take the drug, take the extract. Maybe it can be a plant extract or animal extract or any other natural composition, and give to the patient, and to the patient where it is targeting and how it is targeting, how is curing, all don't know. Is a traditional one. Just give some crude and the symptoms will be reduced. Don't know exactly the disease and it is going how to cure. That is a traditional one. And this the high throughput sequence screening, like the leads, if you want to have the protein as target and the drug, so how they are interacting or not, possible drugs, as I told you, are tens of thousands of drug target candidates, which you wanted to try to the target of your protein where it is causing a disease, and how this is called as high throughput screening, which will take a lot of time, whatever the technology it is developing. So you have to do in vitro study, and interaction of those study need to be analyzed and observe that one. It takes a lot of time. And computational approach, uh, can assist identifying the lead using visual screening and also optimizing the lead towards better potency. So the two of the steps play a major role in drug identification. One is visual screening. That means the target and the drug candidate, or we cannot even call the candidate of the drug any ligand molecules. The first step we can just need to call as a ligand molecule, and whether it is having the interaction or not in silico screening or visual screening. When we start with the hundreds or thousands of molecules, you may end up with few hundreds. And in fact, in vivo, may be more accurate, but that it may take 2 to 3 years. But if you can do the visual screening, which can reduce the 2 to 3 years, and but of course you may not able to identify maybe not as accurate exact as in vitro, but it will give a little bit more number of targets than in vitro. So for example, when you start with the 10,000 molecules of ligands, maybe in vitro you may end up with 100 of them are binding. That is only the binding. But if you do the in silico, instead of 100, you may get 300 or 500, but still that is fine. You can save a lot of time and the cost. And failing early is failing safe, isn't it? And where this structural biology can play a role in other areas than drug designing, it also plays in the food industry. So see, the understanding the ligand, which is having a different, the same taste as the natural product, the same kind, same thing like aroma. So here the target is the sensors, either the smell sensors or the taste bud sensors. And we try to understand which is the natural molecule which is giving the taste. And you can create a molecule which can mimic the same taste that use as a ligand to the target. And that is what the different flavors are synthesizing now in the food industry and the aroma industry. And apart from that, protein enzyme designing and also biomaterial design. And yeah, the next one is the drug discovery. There are two different ways we do that: structure-based and ligand-based. Structure-based drug discovery are like a docking, visual screening, or de novo design. And ligand-based are like QSAR and pharmacophore-based. So we will try to understand what is meant by structure-based and ligand-based. For example, structure-based in the sense, you know the target which is causing the disease, where you need to target, that is a target molecule. Most of the times we will discuss that is a protein. Once you know that structure, the next thing what you need to do is you target that structure of the protein. So that is the structure-based. You know the structure of the protein which need to be targeted. So once you know that, your drug discovery is that you need to find a molecule, a ligand molecule, which can bind to the structure of the protein. Okay, that is one way. So ligand-based drug discovery is that you know that the possible drug, which is working previously. Okay, but crudely they used to use it, but you need to find a efficient drug than that. And but you don't know what is the target, where it is going to bind the target, you don't know. But you want to do that. You have a ligand, and based on that ligand, you want to design another drug which can efficiently work. So that is called as a ligand-based drug discovery. Quantitative structure activity relationship is called as QSAR or pharmacophore modeling. So when I give an example of pharmacophore modeling, you will understand what is meant by exactly ligand-based drug discovery. So this target-based or structure-based, receptor structure known. Our receptor, the protein structure we know already. Okay. Mechanism is also known because the target what we know. The ligands and their biological activities are known or unknown. So the which is naturally binding the molecule, that ligand, whether we know or we don't know, that is we can call as a target based drug design, because you know the target where it need to be targeted your drug based on that. The ligand based one, we know the receptor, the receptor structure we don't know, where it is binding we don't know, and mechanism how it is the ligand is going to bind to the protein. Sometimes we know, sometimes we don't know also. But ligand and their biological activities are known. The ligand we know what actually naturally it is binding. It's acting against the particular target, but we don't know the target. But it is actually giving that kind of therapeutical property. But based on that, we are designing another drug. Let's say okay, I'll give you an example. So these are the table where ligand unknown and ligand known, and receptor unknown and receptor known. So come with the first one, which is receptor unknown and ligand unknown. So receptor and ligand both are not known. So that imperial approaches random screening, combinatorial synthesis. So this comes under our traditional screening, where receptor unknown. We don't know where it is getting targeted. Ligand also don't know. What is the molecule we are using? Both don't know. Just giving a crude extract of the plant-based material most of the times, and give as a drug to the patients based on the symptoms. So that particular patient is suffering from some particular symptom, they just give that one. So where both we don't know where it is going to be bind. We don't know what molecule is going to bind. Also we don't know. That is empirical approach. So that is still many places. There's still some of the places we are still using that based on the symptoms, a particular kind of drug we are using. But the which exact molecule and where it is binding, exactly what mechanism leads to be a possible drug of that, also we don't know. Okay, so that is the one approach. Okay, this actually it's at this moment, at present, it's not very safe, because it is having a lot of limitations. How it works we don't know. Then it is a many things, it will be having a limitation. So then ligand unknown and receptor known. That means target but ligand, which is naturally binding molecule to that protein, we don't know. Okay, so then we go for the de novo structure based drug design. We can design a molecule which can act as a ligand based on the protein receptor. So let's say this is a protein and we have a groove here, so that we know the structure of it. So we can create a structure which can bind to here. So that is how it works. Receptor, we know where the active site we know, but what molecule binding we don't know. So we can create a structure. So that is called de novo structure based drug design. Ligand known, but receptor unknown. So that means we know the which molecule is binding to the receptor, but we don't know the receptor structure. So then we go for the QSAR, pharmacophore modeling or database screening, searching. So this we will see in the next slide, how it works. And de novo structure-based or docking, we go for the receptor known and ligand known. So we know the structure of both, like receptor structure we know and ligand structure also we know. So then we go for the binding, docking and see whether it is bind in silico or not. If it is binding, I put in a list, a probable candidates list or the hits list, okay, in the preliminary hits list, which can again confirm by in vitro models, and then we can consider that as a hit. So that is what we do, docking, if we know the structure of both ligand and the receptor. So this is what I was trying to say, pharmacophore. When we do the pharmacophore, when we know the ligand structure, but we don't know the receptor structure. Pharmacophore is the group of atoms common for active compounds with respect to the receptor and essential for its activity. So instead of theoretically, I'll just explain this an example. Let's say this is the procaine and cocaine. Both have anesthetic property. QSAR studies indicate that pharmacophore binding site is related to the presence of, this is the ester bond. See, this is the ester. Also here the ester bond. This is the amino group. This is the amino group. And here the aromatic chain and group is the aromatic group. So this is called as pharmacophore. Here indicates not only the presence of same functional groups, but also the presence of same position. So the same position, first it aromatic, ester, amino and amino. So this kind of the structure you try to mimic and find the other molecule. So let's say this is the cocaine, which is the naturally available molecule in some of the plants, which is having the drowsy and effect. So based on that, you can synthesize another molecule which can give a similar effect and have a drug kind of effect, like anesthetic property. Okay, because that is working on the brain and it is actually giving the anesthetic effect. So you can, based on these two, and again you can find another drug which can have an aromatic ester and amino group and with not much extreme other groups and the similar size and the same position, then you can find another drug. See, receptor map proposed for procaine and cocaine. So where it is exactly this match of Armstrong, where the ester bond is there, and this is the other Armstrong, this is the length, and the amino group, and then a cavity for aromatic ring, and so on. So this is called as pharmacophore of that particular ligand. And based on that, you can design a new drug. Make, that is what usually the pharmaceutical chemist they use, or any, even the normal chemist also. So based on that. So another one is de novo ligand sequence design, where we use the linking growing random connection. So we know the protein structure and the binding site, but we don't know the ligand. What is binding? We don't know. Natural one also we don't know. So we have to synthesize a new one. How to do? So the linking, binding, and docking. So try to do some linking. So it is amino group. So something we need to have a molecule which can have hydrogen bond. So hydroxyl group. And then here some aromatic hydrophobic region. So put some aromatic ring here and then link between each other. So that is the linking. And then go for binding with this kind of ester bond or something which can have linking between the different places where it can have interaction with the protein structure or the active site. So then you can see a docking and this place, take this molecule, free it off and try to do the docking study, whether it is exactly binding this place in the same pose or not. So then you can understand the free energy difference, whether it is lowering the free energy, then it is a possible binding ligand. So binding of a molecule or the ligand to a active site is not enough, that is not going to be a drug. A lot of things need to be screened. So what are those? Selectivity. So drug versus inhibitor. So as I told you, inhibitors alone cannot be a drug. A lot of other things need to be fulfilled, like selectivity. So it need to have a target. The drug should target and bind to the target alone. Okay, it should not bind to the many molecules. It should not have so many targets. When it is bind to the so many targets, which are vital proteins are present in our body or any other animal when you are targeting, so it should not affect that. So selectively bind, only the target it should bind. And less toxicity, so it should not have more toxic. So as low toxic as possible. Most of the drugs are present. What we are using is a toxic, but less toxic. Okay. Bioavailability. So how much time it can able to sustain in our body or in the cell. It should not clear it off immediately, and it should not stay permanently. So it should be able to clear. So the availability should be there in a appropriate time, and that means slow clearance. Slowly it need to be clear, not immediately. Once you take within 10 minutes, it will be clear, then it is not going to be good. So it should be available for certain few hours before it is cleared off by the liver and kidney. So before it is clear, it should be available biologically and reach the target. It should be able to reach. Okay, so like especially blood-brain barrier. So many molecules cannot reach it. So you need to have a signal, certain signal to the drug target if you want to reach the target, especially like a brain. Ease of synthesis. So all it is clear, but it is synthesizing very difficult. It is taking a lot of time. But then it is not going to be a drug candidate. If you cannot able to synthesize easily, which cannot fulfill the needs of the society or the patients, then it is not useful. Low price. So that is rational. Sometimes lowest possible price need to be make it, but sometimes it may not be possible. So lowest possible price need to be the way need to be found. And slow or no development of resistance, especially in case of targeting certain kinds of an enzyme like antibiotics. So if a small change can lead to the resistance, then it is not going to be useful. Stability upon storage as a tablet or solution. So that is important. Like it should be easily transportable to the whole every area of the world. That is what one of the limitation with the mRNA based vaccines what we are facing now. So it required the freezing conditions. Liquid nitrogen or minus 80 it required, So that kind of thing, it is not that easy to store and also to transport. So as much as possible, better to use as a normal temperature, and also at room temperature it should be stable, and it can able to store for a longer period. Then pharmacokinetic properties. So like how that is absorption, then distribution, excretion, metabolism, all these parameters need to be fulfilled. Then no allergies. Out of all these things, if it is having some allergic reaction, then it is not going to be useful. So that's why Lipinski come up with a rule. It is called as Lipinski rule of 5. But we have only 4 points. The 5 mean it is everywhere it is having the rule with the 5 related ones. Like there are more than 5 hydrogen bond donors. It should not have. Poor absorption or permeation are more likely when. So more than 5 hydrogen bonds, then it is not likely a drug candidate. So here don't confuse. There are more than 5 hydrogen donors. So when the more than 5 hydrogen donors that leads to the poor absorption, then it is not a drug candidate. That means the hydrogen bonds should be, the donors should be less than 5. Molecular weight should be less than 500 Daltons. M log P is over 4.5, or it should be less than 5. And sum of nitrogen oxygen should be less than 10. So then in order to become a better drug candidate. So given 2 biological molecules, determine whether the 2 molecules will interact. Is there is any energetically favorable orientation of the 2 molecules such that one may modify the other function. That is to do the 2 molecules fit each other together in any energetically favorable way. So what is the orientation that maximizes the interaction while minimizing the energy of the complex? So when you are trying to find the 2 molecules, whether it is bind or not, so how we can understand, we need to understand what is the favorable orientation. So there are so many orientations are possible for each molecule. So which orientation is the best for binding the 2 molecules, and whether the energy is favoring these 2 molecules. So after binding, the free energy is lowering or not. When it is lowering, it is more stable. To be able to search the database of molecular structures to retrieve all molecules that can interact with the query structure. So we need to have all the database of molecules and try to work with a structure, uh, whether how many of those are able to bind in silico model. That is come with a molecular docking. Molecular docking is a molecular docking tries to predict the structure of the intermolecular complex formed between 2 or more constituent molecules. And docking is a process of predicting the protein ligand complex in which the ligand molecules interact with the binding site of receptor, where it is binding. And the ligand protein interactions are various types. The interactions are very types like van der Waals forces, electrostatic interactions, or hydrogen bonding, etc. So this selects a drug where it is going and binding exactly. Before binding, the structure may be different, but after binding, the protein structure may be different. So that kind of freedom need to be allowed to the molecules to move while it is binding. Protein docking is an extension of protein folding. computationally very expensive process. It required a lot of steps. What do you call the, you when you are understanding that biocomputation, you see the cycles, right? So where it take the instruction from the program, then it reach to the processor do the uh, no take the action, and it will give the signal to the output. So then it will give again back. So it is keep on making this cycle. So how many cycles? So thus it is very huge number of cycles it required. So that's computationally it is very expensive process. So automatic prediction of docking is an important step toward rational drug design. Docking is different from interaction. It is high resolution analysis and prediction for protein function. So it's not just like a docking means not just like whether the physically it is able to what do you call complement each other to bind. No, it's not like that. It is more than that. So it's a while it's while binding to the area of the surface of the protein or the groove region. So how efficiently can have a binding with the protein, like how many kinds of interaction it can make and make it energetically favorable. So that is the docking. So it required a lot of other uh, things to be calculated, analyzed. Structure-based drug design. Okay, types of docking. There are three different kinds of docking we can have. Protein ligand, protein nucleotide, and protein-protein. Protein ligand docking is a molecular modeling technique where the goal is to predict the position and orientation of a ligand when it is bound to a receptor or enzyme. Protein nucleotide docking is a protein is docking with a nucleotide. So protein ligand is comparatively less complex, but when you want to understand the interaction with the nucleic acid and the protein, it's a very highly complex, because nucleic acid is having a lot of molecules. When you are giving a freedom to the each atom of the molecule to be moved to bind to the protein molecule, and protein also having a lot of molecules, to give a freedom both molecules, that is, it is going to be a numerous possibilities, and that requires a lot of computational uh, calculations. So, and apart from that, nucleic acids are highly charged molecules, and the exact nature of interaction between the protein and nucleic acids is not well understood. And protein-protein docking, if you are trying to understand your target is protein, another one ligand, instead of ligand, another protein, and trying to see the interaction. So this is typically more complex, because both proteins are flexible and their conformational space is quite vast, and sampling of all possible conformations of proteins is quite a challenge. More often than not, proteins exhibit a considerable conformational change in order to bind to its partner protein. So this is an example where active conformation, and this is the active C loop, and this is the inactive conformation. So when this is this conformance can have both, or can have in our body. So the drug can be targeted to the active or inactive compound. For example, when you have an active and inactive in the body and have the equilibrium, so the whether active can be more or inactive can be less, or inactive can be more and active can less at the equilibrium condition. So when you keep targeting the active, so inactive compounds again giving back to the changing back to the active mode, because in order to maintain the equilibrium. So thus, so your drug target can be active conformation or it can be to the inactive conformation. So when you target to the inactive conformants and all active going to be converted into inactive, that is also fine. So that is one, I mean, just example, which is you can see in CML leukemia. So the Novartis, the Gleevec compound actually targeted the inactive conformation. So that this is the first generation drug to treat the CML. So it is giving a positive response to 80% of the patients suffering from CML. So where this target is, the drug is targeting to the inactive conformant of this molecule, the protein. Again, the same thing, HIV-1 protease. So drug are trying to understand which is binding to the HIV protease. So this is how this is the possible drug, the inhibitor. And there are so many, potential drugs you can see in the Nature publications by targeting the same HNN1, the protease, HIV protease. So given two molecules to determine whether the two molecules are interact or not, energetically favorable the interaction or not, so we need to understand different aspects of it. So what is the orientation that maximizes the interaction while minimizing the total energy? So we need to understand different orientations of that particular molecule and which orientation is the better to bind so that energetically it will minimize maximum so that it is more stable. The goal is to able to search a database of molecular structures and retrieve all the molecules that can interact with the query structure. So these are the three things we need to understand. Looking for cavity big enough to hold the ligand. And second one is fit the ligand in a different orientations into binding pocket and calculating the binding energy. So first, protein is there, our target is there. We need to find where is the active site. How to do that? Find the active site. If you don't know, there is an algorithm. We are going to understand how the different algorithms on how it works. And second one is fit ligand in a different orientations. So which orientation of the ligand is the hundreds and thousands of orientations of the ligands are possible. Which orientation is best fitting to bind to the target active site? That is the second one. The algorithms are there. And the third one, in order to understand which orientation is the best, we are talking about energy. Each conformation or each orientation, what is the energy it is minimizing? We have to calculate it. So for these three, we have a different ways of algorithms we do. So we are not going to deep understanding of each one. Just superficially, we try to understand there are different ways are available. We will try to know how it works. That is only very superficially we will be going to understand that. So we are not working on deep. If you want to go into deep it, maybe that itself a big science. Okay. So when you go really research in that area, you can understand much more detail on it. So we just go superficial to it, and then based on that, different softwares we do have to do the docking. Okay. Okay, these are the ones how we can go for the ligand, the binding active site, either literature based or computational based. And these are the ways I'll explain. Then docking algorithms, how we are going to dock, whether we give the ligand freedom to change its conformation or not. So if you don't do, this is called as rigid. When you go freedom, it's a flexible docking. We'll try to see. If it is flexible docking, there are different ways we can give a freedom. So we can understand by stochastic, systematic, and simulation. And these are few examples we can understand. Then go for energy calculation. So by binding, I told you, right? We have to understand what is the energy after binding. So it's different kinds of scoring systems we do have, force field or knowledge based or empirical. So by using this, we can understand, so which is a different ways. Okay, so what are the three we are going to understand? One is what is the active site? Protein structure we need to have, that is target structure. And targets, once you know the target structure, what is the active site? We need to understand. There are different ways. Once we understand the target structure, so ligand, we have different ligands. We are going to allowing the ligand to the binding to the active site. So when it is ligand binding to the active site, ligand can have different conformations. So which conformation is better to bind to the active site to minimize the energy? So that means we need to give we need to have a different conformations to bind to the active site. There are some algorithms which makes the ligand to be oriented differently to give the different orientations. And when it is bind to the active site, calculate the energy to have understanding what is the minimum energy of that complex. So how we can calculate the energy? So that is the energy systems will give you how to calculate. Okay, so this maybe I'll explain to you maybe tomorrow or day after tomorrow. And based on that, we do have the different softwares to do the docking studies. So this is the protein, and the protein is having the active site, and the active site the ligand is binding. See the ligand, two ligands are binding. So one is a natural one and is one other one is a drug candidate. So that is how the neuraminidase binding, which is DANA, is binding natural compound, natural ligand binding to the neuraminidase of this influenza A virus, and the drug target is this one. So binding, I mean, the same active site. So Relenza, the GSK product, the structure-based discovery. So they try to do this binding of this to the same target at the same active site, it giving the same effect. So this is resulted into a drug candidate, drug for the influenza A virus. So that is how you can do the binding assay, the docking assay to find the possible drugs. So there are different the softwares we do have to do this docking, and the one of the most popular one is the AutoDock, another one is Gold, another one is Dock. So what we do tomorrow, as I inviting a lecturer from pharmacy, Professor Ravichandran, he will explain us using the AutoDock how to do the docking. So what we are going to do, we take the protein from the database. So you must be knowing by now what are the databases we do have the protein structure. So protein structure database like PDB we do have. We download the PDB protein from the PDB. and prepare the protein for the docking, and they take the ligand and do the docking. What are the different steps? We do have those we can see tomorrow, and the docking. So once you have a practical knowledge on it, maybe I will explain to you theoretically, which I skipped today. That is active site identification and uh, docking algorithms, and then energy calculation options. So we will do that in a following class. And for tomorrow's class practical, so there are the uh, few files or applications you may need to be downloaded and also installed. So what I do, I'll send those things to you. Maybe by tomorrow you work on it. So some of the molecules I sent to you, and software also. Okay. And the protocol also there, Autotalk protocol. So you step by step, you try to work on it if whatever you can do. And otherwise we can start tomorrow. So if you can do by that, maybe we can minimize the time on tomorrow. Otherwise, it may extend beyond the 11. So you try to prepare yourself to be beyond 11. So maybe we can request Dr. Lee, I think, teaching you medical biotechnology. In case of biotechnology students, I don't know bioinformatics, what is there for you after 11. Metabolomics. Okay. If it is required, we can put a note to both of them, Dr. Prabha and Dr. Lee, that they may need to prepare for giving us another 1 hour time, 11 to 12. I will do that and see if the possibility. What I need from you, you need to just download what are the things which I going to send